Welcome learners. From thumbnail you could have guessed already that what will be today's topic of discussion. But at first, in India, October and November of a year is considered as a heavy loaded festive times. But in this pandemic year, we all need to be conscious enough. I wish all of you a very happy and healthy Pujo. Stay negative, stay safe, and keep learning. I made a video of DNA replication in prokaryotes previously, so please check it out that you can differ the replication process between prokaryotes and eukaryotes. I thoroughly followed the book iGenetics by Peter Russell for making this video. This is a must-read book, so let's learn. Each eukaryotic chromosomes consist of one linear DNA double helix, such as the human haploid genome consists of about 3 billion base pairs of DNA, meaning that the average chromosome is roughly 10 to the power 8 base pairs long, about 25 times longer than the E. coli chromosomes. Replication fork movement is much slower in eukaryotes than in E. coli, so if there was only one origin of replication per chromosome, replicating each chromosome would take many days. In fact, eukaryotic chromosomes replicate efficiently and relatively quickly because DNA replication is initiated at many origins of replication throughout the genome. At each origin of replication, DNA unwinds to single strands and replication proceeds bidirectionally eventually. Each replication fork runs into an adjacent replication fork, initiated at an adjacent origin of replication. In eukaryotes, the stretch of DNA from the origin of replication to the two termini of replication on each side of the origin is called the replicon or replication unit. We can draw basic differences between prokaryotic and eukaryotic replication here. In case of prokaryotic replication, the E. coli genome consists of one replicon of size 4.6 MB with a rate of movement of each replication fork of about 1000 base pair per second, while eukaryotic replicons are smaller in humans for an average of 30 to 300 kb, and the rate of fork movement is about 100 base pair per second. There is another differences between prokaryotes and eukaryotes that replicating the entire chromosome takes 42 minutes in case of prokaryotes, while the replicating the entire genome takes 8 long hours in case of eukaryotes. Here we need to know that the replication fork fuse at the margins of adjacent replicons. The chromosome has replicated into two sister chromatids after eukaryotic replication. Initiation of replication is directed by a DNA sequence called the replicator. Replicators are less well characterized in eukaryotes than in prokaryotes. In the yeast Saccharomyces cerevisiae, replicators are approximately 100 base pair sequences called autonomously replicating sequences. The initiator protein in eukaryotes is the multi-subunit origin recognition complex. The ORC binds to two different regions at one end of the replicator and recruits other replication proteins. DNA replication takes place in a specific stage of the cell division cycle. The cell cycle consists of 4 stages. First stage, that is G1, during which the cell prepares for DNA replication. In S phase, during which DNA replication occurs. And in G2 phase, the cell prepares for cell division, and the last phase is mitotic phase, where the division of cell by mitosis occurs. The initiation of replication involves two temporally separate steps. The first step is replicator selection, in which ORC binds to each replicator in the G1 stage and recruits other proteins to form pre-replicative complexes. Unwinding the DNA does not occur yet. And then the pre-RCs are activated when the cell progresses from G1 to S phase, and then they initiate replication. Now, the limiting replication initiation to the S stage is controlled by proteins called licensing factors. And now these licensing factors are synthesized only in G1 and then move to the nucleus, where they are the first proteins that bind to ORCs to form pre-RC. Other proteins are now recruited and the entire complex begins to untwist the double-stranded DNA. At this point, the licensing factors are released from the complexes and inactivated either by being degraded or by being exported from the nucleus. Eukaryotic cells may have 15 or more DNA polymerases. Typically, replication of nuclear DNA requires 3 of these. So these 3 are polymerase alpha or primase and polymerase delta, and the last one is polymerase epsilon. The polymerase alpha that initiates new strands in replication by primase and making about 10 nucleotides of an RNA primers, and then the polymerase alpha adds 10 to 20 nucleotides of DNA. And the polymerase epsilon appears to synthesize the leading strand, and the rest of this, that is polymerase delta, synthesizes the lagging strand. Other eukaryotic DNA polymerases are involved in specific DNA repair processes. Yet others replicate mitochondrial and chloroplast DNA. There is a special problems of replicating completely a linear chromosomes in eukaryotes because of the telomeres. The replication of parental chromosome produces 2 new DNA molecules, each of which has an RNA primer at the 5' end of the newly synthesized strand in the telomere region. So in case of prokaryotes, the numerous RNA primers in each lagging strands have been replaced by DNA during the normal DNA replication steps. It is noticeable that the Okazaki fragment, that is 5' to the RNA primer, is extended in 5' to 3' direction to replace the RNA primer. And since there is no Okazaki fragment 5' to the primers at the 5' ends in eukaryotes, the same mechanism wouldn't work at the 5' ends. So the removal of RNA primers at the 5' ends of the new DNA strands leaves a single-stranded stretch of parental DNA, which is called an overhang, extending beyond the 5' end of each new strand. So DNA polymerase cannot feel the overhang. So if nothing were done about these overhangs, the chromosomes wouldn't shorter and shorter with each replication cycle. So there is a special mechanism is used for replicating the ends of chromosomes. So most eukaryotic chromosomes have species-specific and tandemly repeated simple sequences at their telomeres. So there are 2 scientists named Elizabeth Blackburn and Carol W. Grader have shown that an enzyme called telomerase maintains chromosome length by adding to one strand, which serves as template on previous DNA replication at each end of a linear chromosome. The repeated sequence in human and all other vertebrates is 5' TTAGGG 3', reading toward the end of the overhanging DNA. So telomerase acts at the stage where a chromosome end has been produced after primer removal with an overhang extending beyond the 5' end of the new DNA. Telomerase is an enzyme made up of both protein and RNA. The RNA component includes an 11-base template RNA sequence that is used for the synthesis of new telomere repeat DNA. The telomerase binds specifically to the overhanging telomere repeats on the strand of the chromosomes with the 3' end. And the 3' end of the RNA template sequence in the telomerase here 3' CAAUC 5' base pairs with the 5' GTTAG 3' sequence at the end of the overhanging DNA strands. The telomerase catalyzes the addition of new nucleotides to the 3' end of DNA, here 5' GGG TTAG 3' using the telomerase RNA as a template. The telomerase then slides to the new end of the chromosome so that the 3' end of the RNA sequence 3' CAAUC 5' as before now pairs with some of the newly synthesized DNA. Then as before, telomerase synthesized telomere DNA and extending the overhang. But the process can recur to add more telomere repeats. In this way, the chromosome can be lengthened by the addition of a number of telomere repeats. Then when the chromosome is replicated using the elongated strand as a template and the primer of the new DNA strand is removed, there will still be overhang, but any net shortening of the chromosome will have been more than compensated for due to the action of telomerase. The synthesis of DNA from an RNA template, it's called reverse transcription. So telomerase is an example of a reverse transcriptase enzyme. So the telomerase reverse transcriptase is the abbreviated of TERT, that is TART. So assembling newly replicated DNA into chromosome is needed. Why? Because DNA is complexed with histones in nucleosomes, and nucleosomes are the basic unit of chromosomes, and there are 8 histones in the histone core of the nucleosome, 2 each of H2A, H2B, H3 and H4. Therefore, when a DNA is replicated, the histone complement must be doubled so that all the nucleosomes are duplicated. Doubling involves 2 processes, the synthesis of new histone proteins and the assembly of new nucleosomes. Most histone synthesis occurs during a stage of the cell cycle so as to be coordinated with DNA replication. For replication to proceed, nucleosome must disassemble during the short time when the replication fork passes. The newly replicated DNA assembles into nucleosomes almost immediately. Each parental histone core of a nucleosome separates into an H3H4 tetramer. That means 2 copies each of H3 and H4 and 2 copies of an H2A and H2B dimer. The H3H4 tetramer is transferred directly to one of the 2 replicated DNA double helices past the fork where it begins nucleosome assembly. The H2A, H2B dimers are released, adding to the pool of newly synthesized H2A, H2B dimers. A pool of new H3H4 tetramers is also present, and one of these tetramers initiate nucleosome assembly on the other DNA double helix past the fork. The rest of the new nucleosome are assembled from H2A, H2B dimers which may be parental or new. Thus, a new nucleosome will have either a parental or new H3H4 tetramer and a pair of H2A, H2B dimers that may be parental new or new. So histone chaperones proteins in the nucleus direct the whole process of nucleosome assembly. So this is all about today's video, and I hope this tutorial helps you. If there are any suggestions or queries regarding this channel's growth or today's class, please put a comment below. Do not forget to like and share. Stay safe. Once again, happy Durga Puja.